I'm going to say a few words about one of nature's miraculous tools called enzymes, and in particular the project OXYMOD, which was a digital life project. I'm going to put this in a little bit of a bigger perspective. Why is my slide not moving? There you go. So the background of much of the work that we are doing, and others in Norway, is the concept of biorefining, where we use non-edible biomass, so byproducts, woody materials that are not being eaten, to convert to useful products. There's many ways to do that, but the key of the whole biorefining concept is that one uses renewable feedstocks, employs nature's own tools, so biotechnology, microbes or enzymes, to process those feedstocks, use them optimally and completely, and closed loops, that is, save nutrients. Don't lose phosphate or nitrogen, so bioavailable phosphate, nitrogen, and so on the way. That's like of the big picture where we are operating in here in my group at NMBU. And of course, nature has many tools, and among those tools are the enzymes. This is a poster showing human metabolism. You cannot read it. You see a detail to the lower right. My point is that there are enzymes in nature to catalyze almost any reaction. That means that a lot of the chemistry that we need to do to either refine biomass, but also generally to produce the things that we are producing in chemical factories today, could in principle be done or catalyzed by nature's own catalysts, which are enzymes, and which in principle would allow much greener processes with less contamination, more precise, lower temperatures, less chemical use, and so on and so on. So enzyme technology is a very attractive technology, but of course you have to have the enzyme produced at an acceptable price to doing the right thing in order to, for example, replace classical chemical industry. Enzymes are all over the place, not only in our metabolism. If you think about biorefining, you don't want to see this when you chop a tree. And of course, in nature, there are organisms that can deal with woody materials. They have the enzymes, they have the machinery to convert woody materials to, in this case, the energy that they need to grow. But we could exploit such enzymes, of course, to do things that we consider useful. This has been one item in the OXYMOD project and in many other projects. This is a picture from Acinta Trondheim showing a collection of actinomycete bacteria. And I'm just showing this because it's a very nice picture. And it shows you, in a way, it gives you a very nice feeling of this is bacterial biodiversity. And all these bacteria produce enzymes doing things. So if you're interested in certain enzymes doing certain things, you might assess collections like this and look for whether some of those species here, each dot is like a species, you could say, simplify it a little bit, so it's a different bacterium. You could look at whether some of these perhaps are capable of doing what you would like to do eventually in a factory. And this is one aspect of also the OXYMOD project. I want to focus in a little bit on something that we have been doing here. And the reason that I focus on it is that there are some very nice roots from very fundamental work to application. As you will see, is a nice example of what biotechnology can do. So these brown threads here, these are cellulose fibrils. So this is what you would have in woody material. It's a gross simplification. Don't bother about that right now. And it has been known for a long time that if you want to break this down, there are enzymes. There are so-called hydrolases. They are very well-known enzymes. In this case, it would be cellulases, cellulose-degrading enzymes that use so-called hydrolytic chemistry, which is very common in biology, to chop these long cellulose chains. You see here, you see a single chain of sugars, and they are being chopped by this yellow thing, which is an enzyme. And what this produces is sugar, glucose, that you then can use to make many different things. For example, biofuels, but also other things. And just as a tiny remark to the previous lecture, of course, going to sugar from these materials is one thing, but these biomasses could also very well be used in material science to make new materials. That's a whole other area that I'm not going to talk about. We should not forget about that. Anyway, in this context, we want to break this down to sugar that we then can use to make things we want. And so we discovered a while ago a completely new class of enzymes which also break down cellulose, but they are these red things here, but they do something different. They go on the surface. They don't have to engulf a single chain, but they go on the surface of the fiber, on the piece of wood, if you will. And then they are able to actually shoot holes in that surface. And to do that, they have to use a chemistry that is much more powerful, but also much more difficult to control. It's redox chemistry. That means there's electrons and things going around. And that was pretty unique when this was found. It was a big discovery and has really revolutionized how we look at biomass degradation, both in nature and in the factory. And I'm showing you all these papers, not only to brag a little bit about our achievements, but also to showing you that this is many papers in very nice journals, that this is a really important topic. It's turned out to be really important. And I also would like to claim that we have been able to stay in the forefront of that field also since the initial discovery. And that has advantages, not only for me as an academic scientist, but also for application in industry. And I want to just say it before I forget it. I mean, there is no point going for innovation if the underlying science is not competitive. That is something I think that many funding agencies forget. But if you have competitive science, you can also do competitive innovation. So these LPMOs, and this is brings me to the OXYMOD project and also to the digital life. Now, don't try to look at all the details of this picture. Cellulose here, there's an LPMO working there, so it's an oxidative enzyme. This is why you see the little flashlight here. It's supposed to suggest that there's another type of chemistry. The point is that this discovery opened a whole new, in a way, insight, because there are many enzymes. If you look at bacteria or this fungi that can break down a spruce tree, they have many enzymes that do so-called redox chemistry. And they use compounds like hydrogen peroxide and other small compounds. They need electrons. For those of you not familiar here, you see an electron moving around. The point is that these enzymes are interconnected. So that the degradation of plant biomass, which is not only cellulose, as you see here, but also, for example, lignin, it's different materials that have to be processed in different ways. But there are connections between the enzyme systems that work on these various parts of the plant cell wall. And it's this systems approach and this systems analysis of how do LPMOs and other redox enzymes interact together. That's what we were addressing in the OXYMOD project, which is also likely why this became a digital life project. So the OXYMOD project is, just so that is said, it was 3 Norwegian research organizations, NMBU, Sint-New, and then we had Vector, a company. We had 2 Portuguese partners in Lisboa, and we had 2 companies, not as active partners, but as, how can I put it, advisors and monitors. And you'll see that was actually not just on paper, it was also in practice. So we have been looking at these systems, enzyme systems, so not one time at a time, but trying to get an integrated view of how this could be done. And then, of course, if you understand this in nature, you will eventually also understand how you can best translate it into factory, where you actually process biomass or byproducts. Now, the project, I'm not going to share much details with you. The project turned to some extent out quite nerdy. This is the title of one of the PhD theses that came out of the project. So it's quite a title. It's not exactly a biorefinery that is described here or a lucrative new process, but you'll see that this is actually important. So we have done quite a bit of enzyme discovery. You'll see one example later. And we have done quite a bit of, it became a bit more fundamental than we thought at the start of the project. But on the other hand, we also made huge progress, as you will see. So this is just an example of, this is probably one of the most nerdy theses that I've ever been supervising, but it's a very important one. And for example, here, this is it's actually also published in the scientific literature, but this is real life. This is the biorefinery plant, the biorefinery demo plant at Boracor, where they run at 4,000 liters, so 4 cubic meters, cost about €500,000 krona a week to run it. And our technology, the technology that has come partly out of OXYMOD, has been used here to develop a process, a cellulose processing process that Boracor basically considers ready for investment after the developmental work that they have been doing. And it's not only because of OXYMOD, but it's partly because of OXYMOD. It's partly because of this nerdy thesis that I was just showing you. So here, this is moving to real life. Another thing that I would always like to bring up is that there is a lot of hype in Norway these days that the Norwegian salmon can be fed with Norwegian spruce wood. Now, let me just say right away, that's never going to solve the feed problem for salmon, because we have not enough trees. But the point is that the technology that makes this possible is not that we have been feeding the cell with something, it's because these enzymes, LPMOs, so the enzyme technology, and also subsequent fermentation technology to produce something such as single cell protein has made this possible. That's what you need to transfer spruce wood to something the salmon can eat. That's 100% biotechnology. So it's important. Yeah. All right, this goes even further. I wrote in my little title, Beyond. I mean, we are doing, and now this is pure blue sky science, right? This is fundamental science. But the goal here is actually very green. The goal here is to make new catalysts to do current large-scale chemical processes more efficient. For example, the selective oxidation of alkanes or even methane. That this may not tell you much, but these are 100 million ton operations run worldwide in the chemical industry. And the ultimate applied goal of this very fundamental blue sky project is to develop better catalysts for that. Let me see. There was someone calling me. Yes, and there is more. For example, these enzymes, we are going to try to make them cytochrome P450 catalysis, which again is for the green production of fine chemicals. And very importantly, these enzymes and the knowledge coming out of a project like OXYMOD also enables us now to do some serious work on how to do enzymatic recycling of plastics. I cannot go into detail, but we have new activities there. And because we are a bit, how can I put that, a bit known because it's LPMOs, we work together with the best. For example, here, the NREL group is extremely well advanced on plastic recycling, both on PET, but also on mixed plastics. A science paper here from last year, where they combine enzyme technology with metabolic engineering. I cannot go into detail. What I want to point out is that the investment made through projects like OXYMOD pays off and is both leading to implementation in existing companies and to new ideas to tackle some of the bigger problems that we are facing. Finally, two examples from concrete examples from OXYMOD. I said we did a lot of an enzyme discovery. This is an example of work done at Syntev together with two of the other partners, or two of the partners involved here. It's a new enzyme for potentially processing of lignin, could also be used in other applications, for example, for decoloring, but I cannot go into detail. So this whole enzyme discovery pipeline that I haven't talked too much about has actually been very successful, thanks to this actinomycete collection. And I want to conclude with something which is rather academic, but also quite fun. It's a paper involving 4 of the OXYMOD partners coming out this year in quite a decent journal, where we have actually been showing that the degradation of wood by these enzymes, and this is, as I said, this goes from the lab to the factory, it can be simulated by using visible light, and we show why that works and how you could do that. And it actually, although this is a bit far ahead, sheds some new light on how lignin, which is another compound in biomass, could be used to harvest sunlight. So lignin could be used as sort of a battery to harvest sunlight to drive processes that we have an industrial interest in, like what we saw from Borg and SU1. And it's kind of it's a bit academic also because it's kind of an original thought, and it's kind of fun to work with this, but it's actually real because there's a lot of lignin there. So if you can use lignin to harvest sunlight to do something sensible, there is of course an applied perspective also there. So those were 2 examples from OXYMOD, and I hope I've been able to show you how we have been moving from fundamental to applied, and how the OXYMOD project and the digital life has helped us to do so.